Thank you, Natalia. It's a pleasure to be here again. We have put together a wonderful program of outstanding speakers to follow me, so I'm not going to talk much about the biology as it is currently being practiced. I'm going to provide a relatively brief historical overview to dovetail with what Professor Oftedal talked about. Now, just to get a feel for the breastfeeding in the man, we have been breastfeeding for a long time. The records would indicate that first hominids and Neanderthals and hunter-gatherers, Stone Age farmers and modern man, they all were breastfeeding. And the modern man introduced some caveats to the breastfeeding, particularly breastfeeding was a very common thing in the first 2 to 6 years of life, and many of people in this audience may remember people were breastfed for the first 3-4 years of life all the time. That was the way to bring up a child, you know. And this was often done without any supplements of mammalian food products, you know. There's also a lot of discussion about breastfeeding in the mythology. If you read historical facts of religious, mythological from the Greek, philosophical discussions in religion, and more recently, since the introduction of commercial products and entertainment, we have been talking about breastfeeding in a very different perspective than we have talked in the past. And then we also developed breastfeeding as a fashion, and where we introduced defined local limits where you can breastfeed and where you cannot breastfeed. So these are some of the interesting things which I'm not going to in any detail, but some things we should keep in mind. Going back to prehistory, to Mesopotamian and Babylonian culture, there are write-ups about breastfeeding in ancient Egypt and Greece, and in the Vedas, and in the Talmuds, and in Islam, in the Middle East. Every part of the world, breastfeeding has been a cornerstone of the uptake and upbringing of the children. And I think the patterns of evolution of breastfeeding have also changed with time over the years. It was an alternative of need. Everyone needed to be breastfed in the earlier period. And if there was a lactation failure or a death of the mother, you had supplemental feeding through wet nurses and what you call alternate mechanisms of nursing. And milk from other mammals was introduced much later on, particularly since we got into the agricultural setting. And I was fascinated with some of the way breastfeeding was looked at and how to improve breast supply of milk. And one of them was in a papyrus in the Egyptian culture was to get the milk supply from a woman for suckling a child if she was not producing enough milk, was to let the woman sit cross-legged and eat fragrant bread of soused dourah. I'm not sure what the soused dourah is, but it's kind of a cereal, I think, while rubbing her breast with poppy plants. And this was supposed to be a very common way of enhancing breast milk production at one time in the man. But I think the evolution of breastfeeding was tremendously revitalized during the Christian church, particularly with the Roman Catholic Church. And there's a story of lactation of the miracle of St Bernard Clairvaux. I don't know if anyone has gone to that little chapel in Clairvaux. It's very close to Champagne region in France. And maybe Jerry might have gone there. And there's a statue, and this picture on the right side here is the St. Bernard. He goes to kneel before the statue of Virgin Mary, and he had a problem. He was not a very smart man. He had some mental problems, and he had also conjunctivitis at that time. So he goes and bends in front of the statue of Virgin Mary, and suddenly the breast opens up, and a streak of milk comes out and gets into his mouth and into his eyes. And lo and behold, St. Bernard became a miracle. He became a genius. he became one of the most influential priests in Clairvaux. And this is a painting which was done by, I think, a Spanish artist in Madrid, in what's a museum in Madrid. And this painting is, I think, hanging there. And that changed the whole attitude about breastfeeding. It became a very decent, dignified thing and a very religious thing to do in terms of nutrition and the breastfeeding for the kids. And this was the way it was looked at. The milk of the virgin kind of paralleled the role of the blood of Christ in the Christian theology. And the miracle of lactation of St Bernard was a reflection of the blood of Christ, more or less, which was spread on St Bernard and made him such a genius. And I think we have changed quite a bit since then. As I showed this picture earlier on, we have much better examples of breastfeeding through our mammalian family, which Professor Oftedal mentioned very briefly. And it's a very highly effective mechanism of delivery of both maternal soluble and cellular factors in the suckling neonate via the transfer of products of lactation from the mother. In addition to that, it has also a tremendous emotional factor, which has become a very important part of the mother-child interaction in the early neonatal period and subsequently in later on in life. And I was hoping Dr. Vanderpeer would be here. He made a quotation, which is in the bottom of this, that besides the St. Bernard's miracle, now we have in the continuum of the gestational period after placenta, the breast milk plays the role of maternal blood by delivering maternal soluble and cellular factors to the neonate. And this may be also the induction of microchimerism. Olaf did not talk about that in other mammalian species, but I think this is something, there's a lot more interest coming into the transfer of maternal products, which might induce a true chimeric relationship between the mother and the child, both through placenta and through the lactational products. As I said, I'm going to go really more in the historical perspective, what I think we have gone through, and we should know that history is a very important part of our life. And a Spanish philosopher once said, if we know where we are coming from, we know where we are going. And I think in the history of lactation and any biological processes, I think we should know where we came from in the introduction of mammalian lactation. 5,000 years ago, milk was a cure for everything. It was a cure for insomnia, fluid filled the belly, piles, infections, sexual dysfunctions. It was also very important for contraception, which has been in fact proven to be very true now. And it was a miracle for everything, more or less, for a long time. But it was really Escherich in 1885 who really started to look at milk from a biologic perspective, and he demonstrated for the first time that milk feeding was associated with changes in the milk flora. And maybe people in the second group will be talking about it in more detail. And Ehrlich in 1892 was the first person who showed maternal immunization with ricin will induce protection in the baby, in the suckling baby. That was probably one of the beginnings of maternal neonatal interaction from a scientific standpoint, which we know about. And then somewhere in the 1950s, late 50s, 59, Gugler and von Moralt, and then our friend Dane Hansen in the early 61s, started to look at it in a more elegant manner and really started to delineate the biologic aspects of human milk and the mammary glands. And this was about the same time we were looking at IgA. Tom Tomasi came up with the presence of different kind of immunoglobulin in the other secretions, and Nana Hansen showed it in the milk. And that was the beginnings of our immunologic approaches to looking at in the milk. Now, what do we have in the milk? I think this is something very important to keep in mind, and I'm not going to go into this at all, and I'm hoping many of our speakers will follow through on most of these areas in some depth as we understand them today. There is a large number of soluble products in the milk. Immunoglobulins, which is the first thing we should talk about, and I'll focus on that most of the time. There are a lot of soluble products from cytokines and other T-cell lymphokines. There are HLA antigens, and there are receptors, TLR receptors, and other factors which are present there. The complement and propyridine. There are a variety of protein-derived peptides which are present in the milk. There's an anti-secretory factor and alpha-lactoglobulin, two things. One of them is called the Hamlet anti-secretory factor. This is Lars Hansen, has been very heavily involved in it. And Hamlet is a product which was thought to have very anti-cancer properties, which Katarina Swanberg has been working on for all these years. And there are many other defensins and lactalbumin. We talked a little bit about lactalbumin and lysozymes. And in addition to these, there are a large number of other products, prostaglandins, there are a variety of hormones, leptins and carbohydrates and oligosaccharides. So we're going to talk about them in some more detail in the second and the third section of this conference. In addition to these numerous soluble factors which we have in the milk, we have a large number of cellular elements in the milk also. We did not talk about cellular elements in the mammalian evolution, but I think would be something worth looking at it. There are a large number of epithelial cells. There are T and B lymphocytes. There are natural killer cells. There are dendritic cells. There are macrophages. There are polymorphonuclear leukocytes. And more recently, there is a lot of evidence to suggest there are progenitor cells and stem cells, which are present in milk in substantial number and which may have a very important role in maternal neonatal interactions. I'm going to focus on some of the work which was done in our laboratories by a number of investigators in the 70s and 80s. So that's pretty archaic, and that's very emeritus kind of a discussion I'm making. And we'll leave to the younger folks to talk about some of the newer things which have come up over the past few years. Now, in addition to the cellular elements which are present from the mother and the child, there are also a variety of environmental factors which are also coming from the mother into the breast milk and coming to the child. And these are bacteria, viruses, parasitic agents. There are maternal drugs and particulate matter. There are many environment toxins, and there are other macromolecules which are transferred from the milk to the baby. Now, we're going to talk about microbiome, and I'll put it collectively together in Professor Walker's session, and we're going to talk about some of the macromolecules, oligosaccharides, other things in Professor Lonadal's session, and I'm going to leave those to them. And I'm going to focus a little bit on the immunoglobulins over the next few minutes. I will focus primarily on immunoglobulins and immunoglobulin isotypes. These are some of the studies which were done in our laboratories by Winnie Ogra, where she started looking in early 70s and late 70s, what is present in the milk in terms of immunoglobulins. And this is an example of what we see in IgA, IgM, and IgG over a period of time, beginning day 1 to about 180 days after onset of lactation. And as you see, IgA is the predominant immunoglobulin throughout the gestation, throughout the lactation period in most people. And IgM is relatively small quantities, and IgG is also relatively small quantities present in the milk. And if one looks at the quantities in terms of percent of total protein of IgG, IgM, and IgA, and the total output within 24 hours, you will see it's very clearly IgA is the single most important immunoglobulin isotype, which you see in the milk throughout the period of gestation. Early on, the colostrum has very high quantities, but it goes down over a period of time. And these immunoglobulins have a variety of antibody activities. I have listed some of them here, and some of you will be talking about more about them in more detail. But you see remarkable distribution of antibody and cellular immunologic activity against many of the bacteria and viruses and some other fungal agents in the milk immunoglobulin isotypes. And we will hear definitely a lot more about them. I'm just introducing these concepts here. Now, once these immunoglobulins are taken up by the baby, what happens to them? Well, I think most of them are excreted in the feces, but a small amount, particularly in the early part of life, is picked up by the babies, and you can detect in the serum. And these are some of the studies with poliovirus antibodies looking at from feeding of colostrum, and you can pick up significant quantities of specific antibodies to poliovirus over a period of time, and after 7 to 8 days, it disappears. So it's a passive acquisition of antibody of IgA type from the mother to the child during the course of lactation. Now, I think I have the wrong slides anyway. Okay. Yeah, we got the wrong slides, but I'll go back here. You see, yeah, we got the wrong slide. Okay. I will tell you here briefly here. I had 2 slides here which I had put in, which shows that the distribution of immunoglobulins in man and other animals is quite different. And this is because the placentation and lactation. Some animals will pick up a lot of stuff through placenta. Well, not all of them. Most of the agriculture animals like sheep, goat, cow, they don't pick up a lot through placenta. Man picks up a lot through placenta. And that's why you can have babies, human babies, without breastfeeding, and they will live without any problem. But if you have some of these animals, particularly sheep and cattle, if they don't get breastfeeding, they will die in the first 5-6 days. They need colostrum because most of the immunity comes through them via the colostrum, at least in the first 7 to 8 days before the closure of the GI tract. So it would seem that there may be different kinds of animals here. I made the ones in the black. This is a picture from Bill Butler. And these are the group 3 animals where you have a lot of it coming from the mammary glands, while the man, IgG, comes largely through the placenta. So it would seem that the IgA probably functions to a large extent in the mucosa of the gut itself, at least in the man. While in some of these group 2 and group 3 animals, a lot of IgG has to come from mammary glands into the blood circulation, into the blood circulation via the process of oral feeding. So that's probably one of the biggest differences between many of the species we know and the man and some of the carnivores and rabbits. Most of the IgG comes through the placenta in those situations. Now, I'm not going to talk about this. The question is, where do these stuff come from in the milk? And I'm going to leave it to my friend, Professor Mistecki, to talk about it in more detail. It is really a reflection of the mother's gut and mucosa, which provides the most important information to the baby. The entirety of maternal experiences in the mucosa is transferred to the baby via the breastfeeding. And I'm sure Jerry will be talking about it in more detail later on. Now, very briefly, I will talk about the cellular components which are present in the milk. And this is, again, some of the studies which were done in the early, late 70s by Ogra and some of my colleagues. And here you see that there's a large number of cells which are coming into the milk from the mother in very early on in the first part of the lactation and colostrum, and then the numbers of the cells drop significantly. And most of the cells, as you see, are epithelial cells, but there's a significant number of T and B cells, which are down here, the straight line here, solid line, and over a period of 60 days or so, they also disappear from the milk. So there's a huge contribution of T cells and other cellular components from the mother to the child. And hopefully, Jerry will talk to us about this a little bit in more detail. And these are the studies which were done initially to picking up T cell reactivity through pH stimulation. And one can pick up rosette-forming cells. Let's see. You see the bottom line, which at that time we didn't know that the best marker we had was rosette-forming cells for T cells at that time. And you can see a large number of T cells and that cells at that time. And if one looks at these cells in the colostrum and the peripheral blood of these babies, one can see there's a large number of peripheral blood cells which will react to stimulation with concavidin A or streptokinase or candida. And the patterns of stimulation are different in the peripheral blood and the lymphocytes. So the distribution is rather different. So there's a selective concentration sometimes in the milk than you see in the peripheral blood of many of these lymphocytes. And this is again with viruses. You see interaction for rubella, cytomegalovirus, and mumps, which was done again by Vinnie Ogra during the period of time. So you see a significant degree of T cell reactivity to specific antigens in the peripheral blood, in the colostral lymphocytes. What's also fascinating that reactivity also seems to be carried, again, probably with protection. This is the studies which were done in babies who were fed colostrum early on, and we looked at their reactivity to PPD. And you see in vitro T cell reactivity to PPD, you see in colostrum and milk lymphocytes, which is in the middle, and the peripheral blood of the mother, which is bottom. And you see at least several of these babies did pick up reactivity against PPD, which was short-lasting. After 12 weeks or so, one did not see much, but you do transfer this reactivity. Now, is this reactivity transferred by intact T cells or lymphokines generated from the T cells in the GI tract from the immunized mother's lymphocytes? Is something we'll hopefully find out during the course of the discussion here. But we also see pickup. This is interferon. If you see babies which are breastfed, you pick up very high levels of interferon in the baby's peripheral blood than babies which are bottle fed. So there's also evidence of lymphokines which are being transferred from the mother to the child via the process of breastfeeding. Now, I'm not going to go through this in detail, but just wanted to make sure there are other oligosaccharides and other soluble products which are present in the milk, and we are going to talk about these in more detail. But they have a lot of functions. They have antimicrobial function, they have anti-inflammatory function, they have pro-inflammatory role, they have immunoregulatory role, and many of these will be discussed in more detail during the subsequent presentations in this session and other sessions on Wednesday. Now, what has become recently available, and I'm sorry I had to include this stuff recently because one of our speakers could not make it, and I wanted to put some of the information about the more recent data on cell types, which we would not be able to talk about otherwise. And some of the people are going to talk their relevance in terms of clinical disease or protection. And there's a very high proportion of extra lymphoid memory cells, particularly CD4 and CD8 positive T cells, which are present in the milk. And they're mostly antigen-specific and activated T cells. They're cells, we have markers for homing And I'm sure Jerry will talk about it more in detail in his discussion. And there are also innate lymphoid cells, which are strikingly similar to T cells, and they provide protection by regulating local immunity and inflammation. There are also large numbers of these cells present in them. And also more recently, there's one of these cells, this mucosal restricted mucosal associated T cells, or MAIT cells, which have been identified in the milk. And as I mentioned, there's a significant content of stem cells of multiple lineages, which has been identified in the milk cells. And these are without MHC class 1 or class 2 antigens present in them. There may be some reason for it. I do not know. Maybe some of you will tell us what they are about. Now, these mucosal-associated invariant T cells have been looked at quite interestingly in a number of studies recently. And they are alpha-beta positive T cells with limited T cell receptor repertoire, and they comprise about less than 10% of the circulating CD3 positive T cells. And they have very high content in the GI tract, in the liver, and the genital tract. And they synthesize a variety of cytokines, IL-17, interferon gamma, and they also produce a lot of cytolytic products. And these are uniquely activated by a small molecule of vitamin B2 metabolism called MR1A, which is very interesting to keep in mind because that molecule seems to be important for some of these infections like Mycobacterium, E. coli, and Salmonella. And they produce this molecule during metabolism of the riboflavin and vitamins. And these cells, these MAIT cells seem to are activated, and they bridge the host innate and adaptive immune responses quite effectively. And this has been looked at quite seriously in a variety of situations. And what seems to be the case that particularly reduced MIT cell responses are seen in a variety of disease processes like tuberculosis, Francisella tularensis, Pseudomonas infections, sepsis, cholera, many fungal infections. And there are also some are MR1 independent infections like viral infections like dengue, and obesity has been one of the factors which has been seen with MAIT cells, and diabetes, and other infections like C. difficile. So this seems to be a very interesting cell which has been identified in the milk, in the colostrum, and may have some important role to play, and I'm hoping one of our speakers will talk about it during the next presentations. Now, as I mentioned, the stem cells lineages which are present in the milk include hematopoietic stem cells, mesenchymal stem cells, neuroepithelial stem cells. There are also embryonic stem cells, and there are luminal stem cells which are important for the mammary gland itself. So it contains a large repertoire of stem cells, and they must have some function. There's nothing useless in human biology, by and large. And this is one example done by one of my, her name is also Natalia. Dr. Ninkina did these studies, and she looked at these breast milk stem cells. They will mature into ectodermal germ layer cells or mesodermal germ layer cells or endodermal germ cell cells, but mostly in mesodermal germ cells. They seem to develop in a very defined manner. Now, what their impact of these cells may be on the host, I don't know. For the man, I think it still remains to be determined. But there are some very good studies in the mammalian system where the potential role of these stem cells has been looked at in some detail. This is again one of Dr. Ninkina's slides. And if you take a baby, it absorbs leukocytes and myoepithelial cells, progenitor cells, immune cells, and a variety of stem cells, and they go into stomach, in the intestine of the gastrointestinal of the baby, and from there, they go probably to a variety of tissues. They may go into the lungs, they may go into spleen, they may go into liver, they go into bone marrow, and there's also some data to suggest they may go to brain. So these cells may have an important role, which I think needs to be evaluated in more careful detail. And the studies which have been done so far, I have summarized some of these here, are the mouse and the sheep, or the mouse and the rat, or the sheep, lamb, and the dyad, or the baboon, and variety of others, and the cells where they seem to traffic from oral mucosal transmission or mucosal epithelium to the Peyer's patches, to the lymph glands, mesenteric lymph nodes, thymus, liver and spleen, bone marrow, to the mucosa itself, gut mucosa, the gastric epithelium, and the brain. It's a fascinating concept that maybe we may have something in the breast milk which could be used in a therapeutic mechanism for human disease or regulation of immune responses in the man. Now, this, what I mentioned, the maternal microchimerism, has seemed to be associated with gut-associated lymphoid tissues, because a lot of these C57BL mice have been looking at, and in these mice, what has been seen that the maternal T cells seem to localize specifically in the Peyer's patches of the pup. What they do there, I think, is something which we need to understand, and maybe Jerry may have some information about this. Maternal T cells found in the neonatal Peyer's patches are mostly cytotoxic lymphocytes, and they have very high levels of alpha-4, beta-7, and CCR9. These are gut homing receptors on them, and very low levels of receptors which were homing for systemic homing or integrins. So there is something special relationship for many of these cells for the gut lymphoid tissue. So what their function may be in the host, I think, is something we have to consider. And it will be interesting to see whether anyone has any data, new data, to look at it. Development and regulation of specific functions of the neonatal and mucosal immune system. Some of it is well known. It does influence development of mucosal lymphoid tissue tremendously. There's no question about that. Whether the maternal microchimerism through breast milk will induce maternal progenitor and stem cells in the neonate and have effect on the development of neonatal effector immune responses or neonatal tissue repair, I think are two possibilities which have been looked at, and there, in fact, there is some evidence to suggest that that may, in fact, be happening in some of these situations. Now, we also know that the maternal milk cells seem to differentiate into alveolar and ductal or myoepithelial cells, as I showed you in the first slide. And that they are probably related to formation of new mammary tissue during lactogenesis in the mom. And they differentiate into astrocytes in the brain. In one of the animal experiments, which are fascinating to see, that they do differentiate into these cells which promote blood-brain barrier integrity in the animals. Whether something like this can happen in the man, I think, remains to be seen. Now, this I mentioned the foster breastfeeding by this was a GFP positive protein they're expressing in transgenic mice. And this, when you transfer these mice, you have transfer of GFP positive maternal cells in the suckling pups and the blood-brain barrier in the blood tissue, brain tissue, which is very interesting. And these cells also differentiate into neuronal and glial cells in the suckling brain. So this may be a breast milk mediated microchimerism, which might happen, in fact, definitely happens in animal systems. Does it happen in the human system? I have no idea. This is something one needs to look at in the context of where we are today. I showed this slide, but I will probably, we know microchimerism can take place through circulation, and it can also take place through breastfeeding. And there are some unique characteristics for this in the breastfeeding-induced microchimerism versus lactation versus maternal pregnancy-induced microchimerism. Unfortunately, I have 3 slides which are not here. But this is one of the possibilities which has been suggested, that these human breast milk stem cells may have a role in regenerative medicine in man. It may be they may exhibit low tumorigenic potential and low ability to induce teratomas. This may be something people have thought about it. They may be suitable for autologous transplantation, something worth looking at. Might be less headache for the recipient. And they may have they may be an important source of cells for stroke-associated pathology in the brain due to differentiation into growth factor-producing cells. This has been tried in animal models. I'm not sure that there are any human data where it has been looked at in any. Maybe Helena might have some information about it now. And they may be able to differentiate into endothelial-like cells which are designed to regulate immune environment in the areas affected by the stroke. This is a possibility which has been suggested in the question of these stem cells which have been present in the milk. So I would like to conclude at this point with what we have seen so far. If we look at the mother and the neonate and what is transferred from the mother to the neonate, we have soluble and cellular factors like immunoglobulins and T cell subset and T cell factors, immune complexes, and immune modulators of tolerance or regulation of immune responses. We have microbiome and virome. We have other protective factors. These are prebiotics, oligosaccharides, glycoproteins, growth-promoting factors. We are going to look at these 2 factors and what I talked about initially in the subsequent discussions during this seminar. But I wanted to show you 2 things which may be relevant to the discussions which will follow my presentation, and that is related to Th1 and Th2 responses for atopic and non-atopic situations where breastfeeding may be an important factor. And you see in Th1 responses are relatively common in non-atopic children early on, and Th2 responses are relatively down in non-atopic children, where the atopic children have a higher Th2 responses. And this may be relatively important in the context of breastfeeding because breastfeeding, by and large, seems to promote Th1 type of responses. And this may be important with subsequent discussions which we're going to have about development of allergy and immunomodulation in the context of breastfeeding, which are going to follow these 2 presentations. The last thing I want to tell you about, this is from thousands of years and more recently, is about fertility and breastfeeding. And this is the data which was collected in 1984, looking at fully breastfed babies, mothers, and breastfed babies early weaning and no breastfeeding. If you look at their pregnancy periods where they have birth intervals, it's very remarkable that fully breastfed babies mothers have much larger birth intervals than babies who are not breastfed. So the concept of contraception related to breastfeeding is really something which has held the truth over a period of time. And I would like to close with this final statement here. And this is from 600 BC. It says, "Oh, those beautiful faces, may the child reared on your milk attain a long life, like the gods made immortal with the drinks of nectar." And then one of my colleagues who unfortunately passed away, Frank Oski, was a pediatrician hematologist. He said, and this is something worth looking at it, imagine that the world had created a new dream product to feed and immunize everyone born on Earth. Imagine also that it was available everywhere, required no storage or delivery and help mothers plan their families and reduce the risk of cancer, then imagine that the world refused to use it. I think this is something which was so remarkable. He died young. He was in Syracuse and at the University of Pennsylvania, a remarkable human being. And this was kind of his hobby, to look at breastfeeding. He was a really hematologist, did some remarkable work on hematology. And I just want to close with this. The young lady who's presented this picture did most of the work in my lab. She was the principal investigator and also happened to be my partner. My wife, she died very young. And there were a number of other people. I had a slide which was lost in the transition. These were people, Dr. Okamoto, Dr. Chiba, Dr. Garofalo, Dr. Goldman, Dr. Randy Goldblum, and a number of other people who worked in our laboratory in Buffalo over the years who contributed to the studies which I discussed very briefly. And I would have liked to close here, but we are going for lunch now. Huh? Any questions? But we'll go for lunch later on. But the next presentation will be why I have the slide here. Our next presenter, just make sure we remember who he is and what he did, and we'll talk about him later on. And that's about it. Thank you. I'll be happy to answer any questions. Yes, Jerry.
Now it works. Congratulations, very nice lecture. I was impressed by that painting which you showed. I think it's Ribera, if I'm not mistaken, a Spanish guy from Prado.
It's Prado, yes, yeah, Prado.
But the interesting point is St Bernard had vision, as you saw, but there is also inscription. Mary told him, monstrate esse matrem, in Latin meaning, I demonstrate to you that I'm mother of the world, of the mankind.
Yes.
But there are many beautiful paintings, milk giving immortality, birth of the Milky Way, Tintoretto or Rubens' gorgeous paintings, where drinking milk should have given immortality, for example, to Achilles and, Trojan War. It's a long story. The important point, Ehrlich's papers, two papers, 1893. One of them, he was feeding ricin, and if you put ricin in the animal's eye, there was a necrosis and so on. But when he fed ricin and then put ricin in the eye, they were protected.
That's right.
That was the first paper. Yes. And the second one was the milk.
Right.
Again, absolutely gorgeous paper.
Yes.
And I strongly recommend that you look at those
Absolutely.
two papers. They are really fantastic. I'm glad that you reminded of them.
That's history. I think history is a very important part of biology. Yes.
Thank you also for this beautiful talk. I just wanted to share with you something I heard recently from a collaborator in Uganda that told me that a practice happening apparently a lot in Africa where people are dying from infectious disease is that the partner, the husband, are breastfed. And they take the baby milk because, of course, it's important the man of the family survives, and that's the best way for them to be protected from infectious disease. So it's not only St Bernard did that. Apparently, it's happening a lot, and it's somehow a problem, of course, for the babies. But then there are priorities in some countries that make that happen. So yeah, absolutely. So I think it's still happening a lot.
Alan?
Ray, I just wanted to point out that the immune globulins in breast milk have another very important function, as shown by Charlotte Kessler in her science paper. It controls the degree of microbiota that are produced and whether they're pathogens or not. So I think it's a very important component.
Yes, I left that to you. I very well know the work. The immunoglobulins are a phenomenal thing. But I wish we had a little bit continuity with the other species who don't have immunoglobulins. And there are many species who have only IgM. I don't know whether they have anything in the milk which is really compensating for lack of IgA. But that's why the function maybe of IgA in the milk at the gut predominantly in regulatory mechanism, including microbiome. Any other questions or comments? Well, we'll have 15 more minutes to eat. Thank you so much.